Good day to one and all present over here. I am happy to see all your bright faces, and I am really happy to see all the young children waiting before the screen to gain some knowledge. Today, children, you all are here not because of the teacher, but because of the science itself. Science is such powerful thing by itself. It is very colorful and very beautiful, and it has all the hidden mysteries of the world. You have to unwind one after another and try to understand it. If you are going to really understand the real essence of it, you will enjoy the aesthetic part of life in your throughout your life. You will be enjoying the aesthetic part of it. Okay? Today, we are going to deal about plant anatomy and plant physiology, the internal structure of dicot stem sunflower. Last episode, we were dealing with the internal structure of root of dicot root and monocot root. You are able to see a beautiful flower here, the yellowish golden color flower, the sunflower. Do you all like it? Yes. So, are you not curious to know what is inside it, what it is made up of, how the cells are arranged in it? Yes, let us see how the cells are arranged in a dicot stem. Here you are able to see a beautiful stem of a sunflower. Now, are you able to see so many epidermal hairs on it? Yes. So, if you keenly observe, you can see all the epidermal hairs on the stem. Now, you are seeing a beautiful ground plan of the transverse section of dicot stem. Here there are three main divisions. One is the epidermis, the second one is the cortex, and the third one is the stele. This is the main part of the ground plan in dicot stem. All the dicot stem will exhibit only these three main divisions, and slightly they will vary in the internal assemblage. Now, you are able to see a beautiful enlarged section of transverse section of a dicot stem. You can correlate the ground plan and the enlarged section of the dicot stem. Here you can see the epidermal hair at first, and it is covered by cuticle, followed by the epidermis, and down below it the cortex region is divided into three major parts. One is the collenchyma cell, and next it is followed by chlorenchyma cell, and the third is parenchyma cell, and down below it you can see the endodermis. The endodermis are single layered and have barrel shaped cell, and internal structure of it, the core of it has the vascular bundle, and above the vascular bundle we have pericycle. Okay. Now, this is the enlarged transverse section of a dicot stem. If you are going to see all the things together, you will be a bit confused, I think. Okay. So, what we will do now, I will segregate it part by part and show you how it is there in the stem, and also what function it has to do with it. Here you can see somebody is peeling the epidermal layer from a stem. Are you able to see that? Yes. Now, this is the enlarged structure of a epidermal cell alone. Here it is made up of single layer of parenchymatous cell, and it is having so many epidermal hairs here and there on it. Now, if you see, epidermal hairs are largely present in plants which are in high temperate region. Okay. In cold region, if the plants are present, they will not show so many epidermal hairs. Then it is covered by cuticle, and the epidermis is made out of parenchymatous cell. The stem hairs largely help to reduce the transpiration. The cuticle over it helps for mechanical support, and prevents injury to the plant, and by and large it is preventing water loss and also infection from the pathogens all around it, and so it is protective in function. It is saving the stem from pests and pathogens. Now, you are able to see the next region, that is the cortex. The cortex is divided into three major regions. Okay. Then these collenchyma cells give mechanical support to the plant, and down below it we can see chlorenchyma cells. What do you mean by chlorenchyma cell? The cells which are largely provided with chloroplast pigment or chlorophyll pigment and helps in photosynthesis, they are called as chlorenchyma cell. Here first we saw collenchyma, it is giving mechanical support, then it is followed by chlorenchyma. The chlorenchyma largely helps in photosynthesis and it is followed by parenchyma cell, loosely arranged parenchyma cell, where large cells we can find very large loosely arranged parenchyma cell with thin wall and intercellular space. Here much of gaseous exchange takes place and they help in storage of food also. It is followed by endodermis. Endodermis has barrel shaped cells and starch granule in it. The starch granules forms interconnected, the starch granules are interconnected and they form a starch sheath. So, this endodermis layer is also called as starch sheath layer. So, now children, can you understand the cortex? Three major regions are there. One is collenchyma, the next is chlorenchyma and followed by parenchyma. The last, the internal structure is constituted by stele. Stele comprises pericycle, vascular bundle and pith. All the three together, the pericycle, vascular bundle and the pith together forms the stele. What is the work of pericycle? The pericycle has alternate sclerenchyma and collenchyma cell along with parenchyma cell, and it helps in supporting the plant, giving mechanical support, and it is followed by the vascular bundle. The vascular bundle has xylem and phloem tissue. The xylem always helps in conducting water and phloem always helps in conducting the food material, prepared starch by the plant to all the parts of the plant body. Yes, so this vascular bundle is very special because it has a cambium in between the xylem and the phloem. If cambium is present in between xylem and phloem, it is a open type vascular bundle. We say it as open type vascular bundle. Why? Because the xylem and the phloem are separated by cambium in between, and this is followed by pith. The pith is made up of all circular oval shaped parenchyma cell which are very loosely packed and they help in gaseous exchange and also to fill in the pith. Yes, large central parenchymatous zone forms the pith. It helps in gaseous exchange and storing the food material. Now, we see the internal structure of a dicot stem sunflower. It has epidermis. To recap, I am saying this, it will have epidermis, the cortex region and the stele. Epidermis is the outermost single layer made up of parenchymatous cell and it has epidermal hairs. The cortex is divided into three regions, the hypodermis, middle cortex and the inner cortex. Outer cortex, middle cortex, inner cortex. The outer cortex is said to be hypodermis. It is called as hypodermis. It consists of 3 to 6 layers of collenchyma cell and it is giving mechanical support to the plant. The middle cortex is comprised with chlorenchyma cell, helps in photosynthesis. The innermost cortex is made up of parenchyma cell and it is helping in gaseous exchange and storage of food. Now, you all will think how to draw this. Usually they ask a diagrammatic question to you and they ask you can draw the internal structure of a dicot stem or a dicot root or a monocot root or a dicot leaf, like that they will ask. Whatever structure you are taking, it is with full of cells and your mind will be not inclined to it. How to go about with the diagram? It will be a very big question mark lingering in your mind. So, I will say an easy methodology to draw the diagram very easily. First, you have to make up your mind such that you have to see which part of the diagram you have to start with and how it has to be followed. Now, you see number 1, I have drawn 2 parallel lines slightly crossed, yes, and the next one I have drawn a curved line to do the epidermal cell over there. Down below it, it is endodermis. So, first you will draw 2 lines and a curved line above that. In picture number 2, you can see that. In the third picture, you can see 2 lines, curved lines, 2 times I have drawn. On the top, it is for epidermis, down below it, it is for endodermis. And then below the endodermis, you can draw pericycle line. Okay, you have to draw pericycle cells over there and then it is followed by vascular bundle in the 5th diagram. This is very simple. If you see the whole diagram together, you will get confused, but if you segregate it into parts and pieces like this, then you can draw it very easily. Within 5 minutes, you can draw this diagram. Yes, only you need to practice it for 2 or 3 times. You can easily catch it up. So, first you will draw 2 parallel lines, then for endodermis and for epidermis and for pericycle. Below the pericycle, for vascular bundle, you will draw the outer sketch. Then coming to the 7th diagram, you can start with the epidermal cell. The epidermal cell in dicot stem has epidermal hairs. So, 1 or 2 epidermal hair you can draw on the epidermal cell and down below it, then you will draw barrel shaped endodermis cell. Down below it, we have drawn an outer sketch for pericycle. Pericycle is comprised of sclerenchyma cell and it is very thick, double layered cell you have to draw here and followed by vascular bundle. In vascular bundle, we have to draw cambium first because here phloem and xylem are separated by cambium. So, below the pericycle, you will have vascular bundle, you will leave some space and draw the outer line for vascular bundle. In the vascular bundle, you will draw cambium at first and then you will fill the cells for phloem on the top below the pericycle. Then below the cambium, you can draw circular cells, the larger cells on the top and smaller cells down below you can draw, and this will be the xylem part of the cross section. Yes, now it is almost complete, is it not? So, only filling up part you have to do now. So, you can see first you see the last diagram. When you see that at first, you will be wondering how I can draw this, but you see the first diagram, what we have started with only 2 lines, then slowly we are developing that diagram very carefully and so we can finish it within no time. And now you have to keep in mind the cortex region. The cortex region will have 3 type of parenchyma cells only. You need not have in mind how I will draw collenchyma, how I will draw chlorenchyma and how I am going to fill the parenchymatous cell down below. Everything is loosely packed oval shaped cell, here and there you have to leave intercellular space and then draw circular shaped cells. And for the first 4 or 5 layers, you have to shade so that it will become collenchyma cell, and then you will come to the chlorenchyma cell. In chlorenchyma cell, we have to put dots to indicate the presence of chlorophyll in that, okay. And then 2 to 3 layers or 4 layers of chlorenchyma cell. After that, you can just draw circular cells, so that it will be filling the parenchymatous layer in it. So, first collenchyma cell, chlorenchyma cell, and then the parenchyma cell. Followed by it, we have drawn 2 lines, curved lines. In that you will be drawing endodermal cell. Endodermis is made up of barrel shaped cell. So, barrel shaped cell you will draw here, and down below it you will draw cells with hexagonal shape, and double layer you will do, okay. You will be doing double layer of cells, and then followed by phloem and cambium, down below it xylem. So, easily you can finish the diagram. Do not think Miss is saying very easily she can draw because she is elder person, how I can do. It is not like that. Simply you follow what I have said you, quietly follow what I have said you. Draw slowly once or twice, and then you can retain all the words in mind also, and within no time you can write the essay type question for this also, no need to by heart. If you have very clearly understood the parts of the diagram, you can easily develop and write a big essay about this dicot stem. Next, we will go with the internal structure of monocot stem. Here we have taken the example of maize. You all like maize, isn't it? Yes. How do you eat it? By frying? Yes, by roasting or steam cooking it. We eat maize happily, but have you ever thought what is inside it? Yes, the stem part of it shows various kinds of cells. Now, you can see in the picture the stem of maize. We are cross secting it, CS of stem. The ground plan is in figure C. So, you can see the epidermis, the ground tissue, and the vascular bundle. Here it is not divided into cortex as it is shown in dicot stem. Here only 3 major things: epidermis, ground tissue, and vascular bundle. Now, compare the diagram of a dicot stem and a monocot stem. In a dicot stem, we have epidermis, cortex, and stele. In a monocot stem, we have epidermis, ground tissue, and vascular bundle. The dicot stem, I told you, its epidermis, cortex, and stele. Now, it is our own jargon to remember the words. It is epidermis, cortex, and stele. It is epidermis, ground tissue, and vascular bundle. Some children will have a patience to write and see. Yes, they will write again and again and retain the words in mind. Some children will sit silently and they will retain the words in mind. Some will be very playful, they like to sing and remember. In my schooling time, we used to sing so many songs with the difficult words so that we can retain it in our mind. Now, shall we try a song with this? Epidermis, cortex, and stele, here dear. Dicot stem shows epidermis, cortex, stele here. Yes, very easy, simple. You sing with your friends so that you can read all the words easily. Again and again you can retain the words in mind. Dicot stem shows epidermis, cortex, stele here. VB, vascular bundle here dear. Yes, for monocot stem you have some other words: epidermis, ground tissue, and vascular bundle. How can we retain this? A simple method. Yes, epidermis, 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 ground tissue, ground tissue, ground tissue, ground tissue. VB, vascular bundle here dear. some music, some song which is lingering in your mind, you can fill in all the words. I am just saying you a sample only. You are all very bright enough to introduce new words into it and learn all the difficult terms like this. Yes. Now, we will go to the internal structure of monocot stem maize. The entire structure you are able to see now. The outer epidermis is not having any epidermal hair. It is only lined by cuticle. Okay. Down below it, we have single layer of parenchymatous cell forming the epidermis, and down below it we can see the hypodermis. The hypodermis, as in dicot stem, we do not have layers of cell. Here clusters of chlorenchyma cells are seen here and there, and it is followed by ground tissue. The ground tissue has vascular bundles scattered over it, embedded in it here and there. The bigger vascular bundle are seen towards the pith, and the smaller vascular bundle are seen towards the periphery. So, this is the ground plan and the entire structure of monocot stem maize, and the vascular bundle is covered by bundle sheath. It is very, very important. The vascular bundle is covered by bundle sheath. The internal structure of monocot stem we will dissect and see. First, the epidermis. Epidermis is covered by cuticle. What is the work of cuticle? It is the outermost layer. It is helping in controlling the transpiration and injury to the plant. There are stomata, so many stomatas, and the stomatas are less in number in monocot stem. The next, we can see the next layer of cell, it is hypodermis and chlorenchyma. The next layer is comprised with hypodermis and chlorenchyma. Hypodermis is made up of sclerenchyma cells interrupted by chlorenchyma cell. So, all the cells you are going to draw here will have double layer. You have to do 2 times. Inside you have to shade, and then externally also you have to draw the outer line. Okay? And the sclerenchyma provides mechanical support to the plant. The internal structure of monocot stem, the ground tissue is shown here. Here you can see the vascular bundle covered by the vascular sheath, bundle sheath, and the vascular bundle is a closed type. Here we do not find the cambium. The cambium is absent here. The ground tissue is the entire mass of parenchyma cell next to the hypodermis and extending up to the core. Okay. So, the vascular bundles are scattered here and there in circular pattern sometime, and the larger cell of vascular bundle, larger grown up vascular bundle are seen towards the center, and the smaller ones are seen towards the periphery. It is not differentiated into endodermis, cortex, pericycle or pith as you see in dicot stem. Again I am saying it is not differentiated. The ground tissue is not differentiated into endodermis, cortex, pericycle or pith as you see in dicot stem. The epidermis is the outermost layer. We will recap very fast. It is made up of parenchyma cell and it is covered by cuticle, and multicellular stem hairs are absent, and stomata are also very less in number. Here the hypodermis is made up of sclerenchyma cell and chlorenchyma cell, and the sclerenchyma cells are providing mechanical support to the plant. The ground tissue, the entire mass of parenchyma cell next to the hypodermis extending to the center is called as ground tissue, and it is not differentiated into endodermis, cortex, pericycle or pith. So, this is all about the ground tissue. Now, the vascular bundle in detail we will try to analyze. The vascular bundle are skull shaped or oval shaped scattered in the ground tissue. Here the vascular bundle are conjoint. What do you mean by conjoint? In the first episode we were trying to analyze what is conjoint. Yes, when the xylem and the phloem are present in the same radii, it is called as conjoint bundle. What is collateral bundle? collateral bundle, and enda condition is also seen here, and it is a closed type vascular bundle we are seeing. Each vascular bundle is surrounded by few layer of sclerenchyma cell, and it is called as bundle sheath. What is the work of xylem here? The xylem always conducts water. It consists of metaxylem. The larger cell of xylem are called as metaxylem, and the smaller cells are called as protoxylem. The xylem vessels are arranged in V shape or Y shaped structure. In mature vascular bundle, the lowermost protoxylem disintegrates to form the cavity. A cavity is formed by disintegration of protoxylem, and this is called as protoxylem lacuna. In the phloem, if you take phloem, it consists of sieve tube elements and companion cell. Here the phloem parenchyma and the phloem fibers are absent. The pith is not differentiated in monocot stem. If you take the pith, it is not differentiated. Other than that, we can find medullary rays. What are medullary rays? The medullary rays appear as radial planar structures perpendicular to the growth of the growth rings. It is growing perpendicular to the growth rings, which are visible to naked eye. These cells cut out by the cambium towards the periphery, and they are phloem. Here medullary rays stores and transport food material. These medullary rays are otherwise called as pith rays are essential, and they are essential for radial conduction of water and mineral salts and organic substances. They transport all the substance from the center to the periphery. Yes, so medullary rays are very important. The rays are also known as vascular rays or pith rays. We will try to see the difference between the dicot and the monocot stem. Now, first if you are going to take the tissue, it is hypodermis. In dicot stem, the hypodermis is comprised with collenchymatous cell, and in monocot stem, it is showing sclerenchyma cell. The crown tissue, if you are going to take next, it is differentiated into cortex, endodermis, pericycle and pith. But in monocot stem, we do not see any differentiation, it is undifferentiated. Now, at third level, we are going to take the vascular bundle. The vascular bundle in dicot stem are very less compared to that of monocot stem, and they are uniform in size and arranged in a ring like pattern, and they show open vascular bundle and bundle sheath are absent. And if we take monocot stem, they are numerous in number, and similarly they are smaller. The smaller vascular bundle are near the periphery, and the larger vascular bundle are towards the cortex. You can see them towards the cortex. They are closed type vascular bundle, and the bundle sheath are present here. And secondary growth is seen in dicot stem, where it is absent in monocot stem. Pith is present in dicot stem, pith is absent in monocot stem, and medullary rays are present in dicot stem, and medullary rays are absent in monocot stem. So, easily when you retain the diagram in your mind, you can write this within no time without any mistakes. Now, it is very easy. If you have an inclination to read this, you will retain all the hard words in mind, and you will enjoy the essence of it. Very happy to have all of you with me, and I hope you have enjoyed the class, understanding it better, and you will perform well in your future. Thank you. We will meet in the next episode.